Today we are going over the differences between DNA, RNA, and protein. DNA and RNA are both made of nucleotides. The nucleotides consist of a phosphate group, a 5-carbon sugar, and a nitrogenous base. The differences between RNA and DNA are found in the 2' spot on the 5-carbon ring. In DNA, the 2' spot has a hydrogen, whereas in the RNA has a hydroxide. This is important because a single hydrogen is much more stable than the hydroxide in the ribose, so deoxyribose is much more stable than ribose. And furthermore, a nucleotide is a polymer, and the different sections of the nitrogen bases inside of the nucleotide have 2 different subsections. There are the purines and the pyrimidines. Purines consist of adenine and guanine, whereas pyrimidines consist of cytosine, uracil, and thymine. Uracil will only ever be seen in RNA, and thymine will only be seen in DNA. There are also a few more differences between DNA and RNA other than just what is found on the 2' carbon. There is a difference in the backbone. They are both held together by phosphodiester bonds, which you can see right here, and that's simply a bond just between the phosphate group and the 5-carbon sugar. And this picture also just further shows that the DNA has the hydrogen on the 2' and the RNA has the hydroxide on the 2'. Furthermore, DNA is a 2-string structure, whereas RNA is only 1 single string. So DNA forms a double helix, where RNA is also, just like I said, is a single strand. In this picture, you can also see that the DNA has thymine, whereas the RNA has uracil. This is an important difference because if you're looking at a single strand in either of these, you will be able to see if it's DNA or RNA on the base pair. So if it's thymine, it'll always be DNA, and if it's RNA, it'll always be uracil. There are specific sequences in pairing in the double strand and the single strand. So with DNA, adenine always pairs with thymine, and cytosine always pairs with guanine. On the other hand, in RNA, adenine always pairs with uracil, and cytosine still always pairs with guanine. The only differences are in thymine and uracil, because you could only see those specific bases in their specific nucleotide. And adenine and cytosine are both purines again, and thymine, uracil, and guanine are both pyrimidines. And you can also see that I have how many hydrogen bonds are shared between each base pair. This just shows the stability. So if there's 3 hydrogen bonds as seen with cytosine and guanine, it is more stable than the adenine, thymine, and adenine, uracil bonds. We're just going to do a little practice with base pairing. So first we're going to do a strand from a DNA. We have to remember DNA is a double helix, so it has 2 strands. Those strands run anti-parallel to each other. So instead of starting in the 5' direction, it will start in the 3' direction and end in the 5' direction. So then we first have an adenine. In DNA, adenine always pairs with thymine. So you're going to have thymine, thymine, because we have 2 adenines, so they'll always base pair to thymine. And then we have guanine. Guanine always base pairs with cytosine, and likewise, cytosine always pairs with guanine. And now we have thymine, so that always pairs with adenine, and so on. So adenine, thymine, cytosine, guanine, cytosine with guanine, guanine with cytosine, and lastly, adenine again with thymine, because this is DNA. So again, these strands run anti-parallel to each other. You have to always remember that, because this will show the difference between the coding strand and the non-coding strand when you get into transcription and translation. Okay, so now we're going to do more practice, but with an RNA strand. You have to remember that RNA is always with uracil instead of thymine. But RNA is only a single strand. So this is just for further practice to show what pairs with what, and that the difference between thymine and uracil. because you'll never see this on the other strand because, again, RNA is a single-stranded. So just for further practice, we're going to go over this. You can pause the video now and fill this out and check with mine in a minute, so I'll give you a second to do that. Okay, so now we're going to pair. So we first have cytosine, and cytosine is always paired with guanine. But then we have a uracil, and that is something we haven't seen yet. And uracil always goes with adenine, because this is RNA. And adenine always goes with uracil, because again, this is RNA. And this is similar to the last one. These are both guanine, so these both go with cytosine. Two adenines, which always go with uracil and RNA. Don't get that confused. Uracil with adenine, guanine with cytosine, and cytosine with guanine. Now this slide just further shows what I started talking about when I spoke in the DNA-based pairing example. DNA has two strands that run anti-parallel to each other. They run in the 5' to 3' direction and 3' to 5'. The 5' end always has a phosphate group attached. So that's an easy way to look at it. If your teacher were to give you a DNA strand and wanted to ask you where the 5' end is, you'll just have to look for the phosphate group that is like this. Because as you can see with the 3', there is no phosphate group. And furthermore, the strands always run opposite each other because it is anti-parallel. Because you have to have the opposite bases pairing with one another. With proteins, you begin with the primary structure. The structure of an amino acid consists of a amino group, which is the NH3+, and a carboxyl group, which is a COO-. These are attached to a carbon, which is attached to a hydrogen and an R group. And the amino acid is the basis of a protein. The protein is made up of a sequence of amino acids. Firstly, in the primary structure is when you see the sequence, so it is just a straight line in which protein amino acids connect to. So imagine these lines are just amino acids. There's no folding. It's just a bunch of different R groups that attach right here to the amino acid structure. So this amino acid structure is the same for all of them, just with a differing R group. And for the sequence to occur, for the different amino acids to bond to one another, the carboxyl bonds to another amino group in a different amino acid thing through a peptide linkage. And then for an amino acid sequence, which is the primary structure, you have 20 to the n possibilities because there's just so many different amino acids and you can form them in so many different ways. So your protein primary structure can be huge because there's no folding and it can be super long and make whatever it wants to be. So again, it's just a sequence of amino acids. And there's 20 amino acids, which is seen in this picture to the left. You have polar side chains, non-polar side chains, electrically charged side chains. And these just vary in how the structure will fold. But it doesn't fold in the primary structure. Again, the primary structure is just a sequence of amino acids. Following the primary structure of a protein is the secondary structure. The secondary structure is made up of an alpha helix and a beta pleated sheet. The alpha helix is a right-handed coil that results from the hydrogen bonding between nitrogen and hydrogen groups on one amino acid and the carbon double bonded to oxygen groups on the other. On the other hand, the beta pleated sheet is when two or more polypeptide chains are aligned and hydrogens form between the chains. So, the important thing to remember about secondary structure is the hydrogen bonding that stabilizes them. The secondary structure is the tertiary structure, and the tertiary structure is the folding that results in a macromolecule with a specific three-dimensional shape. And the outer surfaces present the functional groups that interact with the other molecules. Specifically, the hydrophobic groups, the ones that do not like water, will fold toward the interior of the protein because they don't want to be by water, and the hydrophilic amino acids will point towards the outside. And hydrophilic amino acids consist of the positive charged ones and the polar ones because they want to interact with water, which is also polar. So again, and lastly, there's a quaternary structure in proteins. And the quaternary structure results from the interaction of the subunits by hydrophobic interactions, van der Waals forces, ionic bonds, and hydrogen bonds. And each subunit has its own unique tertiary structure. So the quaternary structure is made up of a bunch of different tertiary structures. So you can see with this picture that all of the different colors are different tertiary structures. And those tertiary structures come together to form the quaternary structure, which is, again, just structure-function relationships. So this shape is an advantage then if all of these different tertiary structures were to be alone.